Our next speaker is Dr. Dennis DeLugos of Children's Hospital of Philadelphia. And what you're going to hear from with Dr. DeLugos is the latest in terms of what they're doing with precision medicine clinical trials. And I know that's been of great interest to everyone. Join me in welcoming Dr. DeLugos.
Thank you. Good afternoon. So what I'm going to talk about is hopefully a glimpse into the future. Imagine that there is a drug that is being tested in a clinical trial for KCNQ2. What will that look like? Now, the interesting news I have is there are very recent examples in other pediatric neurological diseases of how these trials will unfold and how the FDA will react to them. And there are a few things that the community is already doing and can continue to do to try to grease the skids to make the approval process quicker and easier. So we're going to start with some news flashes about precision medicine, not in epilepsy, but in other diseases. And then we'll talk about what that has to do with KCNQ2, and then how the FDA works in general, and then how a future trial, what a future trial might look like. So here's the news flash. There's two news flashes. These are recent headlines from about 3 weeks ago. Approving a muscular dystrophy drug ignites a civil war at the FDA. And FDA okays first muscular dystrophy drug. Await proof it works. So what's that all about? How did they approve a drug without proof that it works? Well, here's how that worked. So a quick, very basic primer on muscular dystrophy. It affects boys. It's a muscle problem. So they are delayed in their walking and they ultimately stop walking, and life expectancy is about mid-20s. This is normal muscle, and it's stained for a protein called dystrophin, which is a normal part of our muscle. And this is a muscle biopsy from Duchenne's muscular dystrophy, and you see there's very little dystrophin. So the problem is there's a problem with the gene that encodes dystrophin, and there's not enough dystrophin, and muscles don't work properly. There are many different types of mutations, but some of them, about 13% of kids with Duchenne's. So a minority, 13%. The patients are amenable to a treatment that involves something called exon skipping. So a quick genetic primer. You guys are pretty good at this already, but this is normal, a normal patient. And these are the exons that make up the protein dystrophin. And notice they fit together like pieces in a puzzle. They tightly fit together. So these are patients with deletions in the dystrophin gene. In this case, exon 50. In this case, exon 49 and 50. And with those deletions, the puzzle doesn't fit together. So you get a shortened, abnormal, dysfunctional protein. Well, if you, in a sense, expand the deletion one exon, you skip exon 51, then the puzzle fits together again. You get a longer protein that's not normal, but it works better. So that was the idea, and that was many years of biology and animal models, understanding how this works and how skipping this exon might help. Now here's the data from humans. Now notice the numbers of patients. 10 patients. The outcome measure is a 6-minute walk test. And this was one of the studies that led to approval of this compound. So here you have here you have months, and it goes from, I think, 12 up to 48 months. And this is how long you walk in 6 minutes. So here, the purple line are patients that got the drug right from the start. And this gray line are patients who got the drug, I think, 24 weeks late. So in the trial, everybody got the drug. It was just a question of whether you got it early or 24 weeks later. And at the end of the trial, those that had gotten the drug from the beginning walked longer. I think it says 67 meters longer over 6 minutes. So that's a 10-patient study with a start and then a delayed start that resulted in a positive outcome. Now, in addition, since muscular dystrophy, you can do muscle biopsies. Now, KCNQ2, you're not likely going to do brain biopsies, but in this condition you can, and you can measure the amount of protein. So the purple line is the amount of dystrophin, this muscle protein, after 48 weeks compared to 24 weeks. So the longer you're on it, the more dystrophin. And then 48 weeks versus 12 weeks, and then 24 weeks versus baseline. So however you measure it, there's more dystrophin. So another biological sign that the treatment is working. Now this is the last bit of data that may be the most relevant to KCNQ2. They had, in muscular dystrophy, what they called historical controls. Patients that had been carefully followed using this same outcome measure, the 6-minute walk test. So these are the historical controls. So from baseline to 36 months later, they started off walking 350 meters, and at the end of 36 months, walked 100 meters. So a lot less. So that's the natural course of the disease. And then when treated with the drug, over the same amount of time, they walked about 270 meters. So that was also submitted to the FDA as evidence of efficacy. And 12 patients. So all of this work was done in 12 patients being treated with the compound. So as a result of all of this, last month, the drug, which is called eteplirsen, was granted what's called accelerated approval by the FDA. So it was approved for use. Accelerated approval. That means more studies must be done. So the FDA has accepted it works enough, there's enough data that will approve it, but the company must do more studies. Now the good news is, because of this, the company now has funding to survive. The FDA can withdraw the drug if future studies don't show a benefit. Now, this is a challenge with all of precision medicine. Let's say most of precision medicine, is the drug only has an effect in 13% of Duchenne's patients. So precision is just that. Not everybody might be eligible, or not everybody might show an effect from the next precision therapy. And the estimated cost is $300,000 per year per patient. So this approval is what created the civil war within the FDA, because there was a lot of controversy over whether this drug met the usual standards for effectiveness. Had enough studies been done? And in very narrow decisions and with lots of controversy, they granted the accelerated approval. The other news flash in nerve and muscle disease is literally from this morning. There's a nerve and muscle meeting going on in Spain right now as we speak. And as I was checking the web this morning and thinking, should I update my talk, I came across this news flash from 5:51 a.m. this morning. About a different disease called spinal muscular atrophy. Now, this is a disease that presents in babies in its most severe form, and the babies are cognitively normal, but they're very weak. They don't roll, they don't sit, they don't walk, and the life expectancy is maybe 2 years. So it is a very tragic disease. The biology is pretty well understood, and there was data presented this morning in Spain about one compound called AVXS-101 and another one called nucericin. And they call it an early data win for these two compounds. So I'll tell you a little bit about what we know about these two compounds. So the AVXS-101 is gene therapy. There was a question earlier today about gene therapy. The problem with spinal muscular atrophy is in this gene called SMN1. It basically is out. It's deleted. So one compound uses a viral vector to insert a functional gene into humans. The other uses a different technique where they modify the way the RNA is spliced to produce a better functioning but not normal protein. So two different techniques from two different compounds. As a result, one of them, nucericin, this is the one that affects splicing of the RNA. They have safety data, dosing data, and they actually did a controlled trial with placebo that was stopped early because of efficacy. And the outcomes were motor outcomes. The kids rolled, they sat, Some even walked, and that never happens in this disease. So they stopped their efficacy trial early and have gone to the FDA asking for an accelerated approval. The other thing that is happening with this compound is the company has announced that they plan to start a compassionate use program where the drug will be made available free of charge while this approval process is ongoing. The rub is the drug is delivered by a spinal tap. It's not a pill, it's not an IV, it's a spinal tap. So a whole nother series of questions have to be answered about, well, how does that happen? Thanks for the free drug, but will insurance pay for the spinal taps for this unproven drug that's still undergoing the approval process? It's almost like a game of chicken. Who's going to say no to this in this very tragic disease? So this is a story that still has to unfold. And then the other one, the AVXS 101, they have safety data and dosing data. And what they presented in Spain this morning by the news flashes is that some of their early efficacy data was so compelling in their safety dosing trial that they're going to try to move forward as quickly as possible. They've already been granted what's called breakthrough therapy designation by the FDA, which allows them an accelerated everything. So the process can actually work, where biological knowledge is translated into therapy and wheels start turning literally so fast that nobody knows how to keep up with this. But we'll see all of this play out in the next few months, and I'd encourage you to follow the news on these two compounds, because this, I think, will be the path that we're going to go down with epilepsy in the future. So what does all this have to do with epilepsy? Well, it means that these treatments arose from a detailed understanding of the disease genetics and biology, very detailed. As I think one of the speakers said earlier, the brain is more complicated than muscle. The brain is more complicated than the nerve cells in our spinal cord that control a lot of our movement. Those are pretty complicated, but the brain's a whole lot more complicated. So there's no substitute for understanding the genetics and the biology. The other news is that pivotal trials acceptable to the FDA can be small, very small. The clearest way to get any drug approved is with a placebo. And one of those, most of the compounds do undergo at some point some kind of placebo trial. But there are some other ways that can be informative and maybe even accepted sometimes. Historical controls. So knowing how KCNQ2 presents without precision therapy is very important. So for example, if the newly diagnosed course is clearly codified, a newborn presents in a certain way, and then seizures unfold, and seizures fail to respond or partially respond at 2 months, at 6 months, at 12 months, could be critically important. And the organization, the community is well ahead of the game, most other epilepsies, in sorting that out. So keeping that work going is extremely important. Having a clinically relevant biological marker like the muscle biopsy. Now that's going to be harder to do, but we'll see how the biology unfolds. And then a lot of the controversy surrounding the approval of the muscular dystrophy drug was the perceived influence of the patient caregiver lobby groups on the FDA. Muscular dystrophy is a terrible disease. Spinal muscular atrophy is. Epileptic encephalopathies due to KCNQ2, they're all bad diseases. Did the lobby groups affect the FDA's decision? Well, from the point of view of those of us in the room, okay, maybe. Let the process unfold. So lobbying helps, and we'll see how the trials unfold in the future. So let's talk about KCNQ2 and a future precision medicine trial. So this is a general slide about what makes a type of epilepsy studyable. There are a few things that make epilepsy types easier or harder to study. This is a list of things that make it easy to study, and many of these things are not good for the patients, but they make it more straightforward to study a treatment. So a striking, consistent clinical presentation with a typical clinical course. So the data that John and others have put together on that neonatal presentation, the EEG, the seizure types, the seizure burden, that's very important. Now there are outliers in KCNQ2, and those exist, but to focus the work on the striking clinical typical presentation is one way to set up the future trials. Having available genetic testing, you have that. Hopefully turnaround time can be even faster. There is talk among commercial and academic labs about having a actionable epilepsy genetic panel. Where a genetic diagnosis could directly inform treatment. So rather than looking at 100 genes, maybe you look at extra fast, you look at 5 or 7, where early treatment could make a difference. So quicker turnaround for genetic testing. In a admittedly twisted way, it helps in epilepsy studies to have very frequent seizures. It also helps to have the seizures be short, basically lots of short seizures. It just makes it easier to design the studies. To have a clear EEG marker. So if the seizures are frequent and obvious on EEG, that can make study design much easier because you can blind the EEG. You have a baseline EEG for, say, 24 hours. You have a post-treatment EEG for 24 hours. The reader doesn't know which one is which, so they're not influenced by what Dr. Goldstein talked about, the natural hope we all have that things will work. You're blinded. You don't know whether it's pre or post-treatment. If the treatment works, if the seizures are frequent enough and the treatment works, that can lead to a much more efficient study. And it helps, again, in a very admittedly twisted way, to have no effective therapies, 0. Spinal muscular atrophy, the disease where the children typically live to maybe age 2, nothing works, no therapies. So wheels turned very quickly in every way as the products were developed and tested in humans. So we'll come back to that point a little bit later. Actually, we'll come back to that point right now. So for future trials, the next compound, is the news about Potiga good news or bad news? Well, if you're one of the patients where it's working, it's clearly not good news if it's discontinued and no longer available. But any type of epilepsy, any sort of disease, if you have several partially effective therapies, incremental therapies, then in a way it makes a precision trial a little more challenging because you have to try this, you have to try that. Those might work. And it delays things, and then it can be harder to study the diseases early. So it's a double-edged sword, however the ezogabine story unfolds. We need a better treatment, a more effective treatment, and simpler path to the study if there is no standard of care, if nothing can honestly be recommended that must be tried first before the new compound is tried. All right, so let's take a step back and look at the FDA approval process, because several speakers have referred to it, including me. Just want to take you through it briefly. What's involved in bringing a drug to market? So for the typical road, let's say this is a drug for hypertension or depression, not rare diseases. Phase 1 studies, healthy volunteers, roughly 20 to 100 healthy volunteers who don't have the disease, and you get safety data and some data about dosing. Then you move to phase 2 studies, and maybe, on average, maybe 70% of drugs make it here. You saw much less for neurology drugs. But phase 2 studies, you're now studying patients. And in a common disease, there's a few hundred patients studied. And this is your first look at whether the drug works, and more evidence about side effects. And then you move on to phase 3 studies, which are much larger, and these are usually the pivotal trials where you have control groups and placebos and rigorous blinding, and this is how you go from phase 1, 2, and 3, and you're into the hundreds or thousands of patients. Now this obviously will not work for rare diseases because no one has hundreds or thousands of patients. So the Orphan Drug Act was passed in 1983 to facilitate the study of rare diseases, and the European Union has a similar designation. So the Orphan Drug Act gives tax credits, other financial benefits for companies to develop drugs for rare diseases. There are many categories where orphan drugs are being studied, including neurological diseases. And there are examples of drugs being approved with very small studies. So Carbaglu, which treats a metabolic disease, was approved based on a case series from fewer than 20 patients, and it was compared to a historical control group. And there's a number of other non-epilepsy products that have been approved with very small trials based on the orphan drug designation. In epilepsy, we have a number of orphan drugs: felbamate, rufinamide, clobazam, ACTH, vigabatrin. Even Diastat is an orphan drug. Fosphenytoin is an orphan drug. So it doesn't have to be a super rare disease to get orphan status, but a rare disease is enough. But there are limitations to this. And we have no approvals right now for very rare epilepsies under the orphan drug status. None of the approved drugs for epilepsy are good enough, whether they're approved for orphan status or not. There's a concern that the Orphan Drug Act has been so good that it has decreased incentives to study common epilepsies. And of course, the treatments are expensive. So there is a path that allows for quicker approval of drugs for rare diseases. So for KCNQ2, what might the first couple of human studies look like? Well, it starts with, unfortunately, we have too much of this, patients with treatment-resistant epilepsy, where the seizures don't stop with available medications, and then caused by confirmed mutations in the same known gene. We have that. The registry is a wonderful example of how patients, academicians can come together to gather patients with mutations in the same known gene. And then, as other speakers have talked about, with lab data, that the mutation is druggable. So preclinical data that you can correct the mutation. And then you need to bring the drug to study in humans, and you need some data on safety and dosage. So maybe the first study would be something like this: 5 to 10 patients who were having very frequent seizures despite standard treatment. Now, KCNQ2 is extra tricky because it has phases. The neonatal phase, then seizures remain, often frequent, till age 2, 3, but then they decrease with age. So you might imagine, would the first study for KCNQ2 be in newborns, or in 2-month-olds, or would it be in 2-year-olds? Might be a different design, different questions involved in that. It probably couldn't be a study of 2 months to 10 years. Too many different phases of the disease in that. So you'd have to figure out what age range, what phase of the disease you want to study. But there would be a baseline phase, and I think the most efficient way is you count seizures clinically and you confirm them by EEG, and you do a developmental assessment, and then this early study would be open label, probably no placebo. You would do some kind of dosing protocol, and then you would do a follow-up seizure count and follow-up EEG, and you'd look for a signal of efficacy. So the outcome would be seizures would be recounted by clinically and by EEG, and a developmental reassessment as well. Let's say you get a precision response. Whoa, it actually works really well. And let's call that an 80 to 100% seizure reduction. Now, in discussions with clinical trialists and funding agencies and other experts, to say, well, you know, big difference between 80 and 100%. 100% is easy. That's an easier study to design if you get 100% seizure reduction. Great, easier said than done, but so the most precise therapy would be no seizures. That's the most straightforward design. If you get a big reduction, that's more straightforward, but it still gets tricky. The trickiest scenario would be you get a partial response. 50% seizure reduction. That's good, but that's difficult to prove conclusively. the ease of proof is easier the bigger the treatment effect. So precision therapy really, I think we really mean precision therapy. It really should work very, very well. And then if you have no response, you move on. And another curveball could be a mutation-specific response. You get a whopping response to certain mutations, but nothing or inconsistent with others. And if that's the case, then you might have to just sort of go back to the drawing board and say, okay, what do we do with like the Duchenne's example? 13% of patients with muscular dystrophy have now an FDA-approved drug. 87% don't. But good news for the 13% who do. And lots of lessons learned for the next muscular dystrophy drug. So if you get a whopping response on these early studies, then you lobby with the FDA to say, hey, our natural history study, our historical control database says these patients are never seizure-free from 2 months to 4 months. And here we have seizure freedom. Grant us an accelerated approval. We'll do more studies, but let the drug be approved. That's essentially what happened with the muscular dystrophy drug. If you get a partial response across all mutations, that's tricky. That's where that can be hard to prove conclusively. And if you get a whopping response with some mutations and not much with others, then you go back to the drawing board and you figure out how do we proceed with patients with certain mutations. And of course, all along the way, you're studying safety, tolerability, and developmental changes, improvements, hopefully. So that's how the FDA looks at things now, and based on the Duchenne's and spinal muscular atrophy, very recent examples, you have a glimpse of how they're assessing those treatments. So final thoughts, all of the science, all of the biology you've heard, they've actually opened the door to these precision trials. Planning in a theoretical way is underway. From a clinical trial point of view, these are the exact same questions, whether it's KCNQ2, KCNT1, any of the rare epilepsies, the same questions come up, which in a way makes it easier. There's a template, hopefully, that will develop for studying new treatments. There already is, and your group has led this, justification for early genetic testing. I think we have a pretty wide clinical consensus that if you have an infant with no cause for their seizures and a very high seizure burden, then you should do genetic testing. And we need to take it up another notch and make that quicker turnaround and a focus on actionable genes. And then again, this room, this meeting, the organization is a model of how patients, parents can all come together and be critical partners in moving this forward, just like the families of the muscular dystrophy patients and the families with spinal muscular atrophy. So that's all I have, and thank you to all of the people that make this meeting and progress happen, and be happy to answer questions. Yes.
Dennis, great talk. I had a question about if seizures become difficult because of some of the reasons you mentioned to look at as an endpoint, what are some of the other ones this community should be thinking about? What seems to have worked in other pediatric severe conditions that this group could start looking at amongst the kids?
Yeah, we talked a little bit about that on a conference call on Friday. And there's two ways to go about it. One would be, there are standardized typical developmental screening tests that are well validated, and they look at your typical developmental progress. The Bayley scale of infant development is one of them. The other is, are there specific developmental things that are different in patients with KCNQ2 mutations? And the example that we talked about on Friday is Rett syndrome. There are some compounds being studied for Rett syndrome where seizures are one endpoint, but there are other behavioral developmental endpoints that are measuring global, development in general, but some specific Rett syndrome phenotypes, autonomic instability, and some typical motor differences, movement differences in patients with Rett syndrome. So if there are fairly stereotyped things that can be tracked and measured in a reliable way, that would be something to look at beyond seizures, because it's hard to get an approval. It's hard to show an approval for developmental improvement in general, especially if you're going to do a 2-month or 4-month or even 6-month study. But if there are specific things, then that might help. Yes.
Last year at the conference, we heard about the SOC4 SF34.
Mm-hmm.
Do you or does anyone else know where in clinical trials that is right now?
I do not. Anyone else in the room? So.
I think the last I heard was that they were intending to go into phase 1 trials maybe sometime this year, early next year.
Yes.
A question on your point on Fatiga, and is that good news or bad news? The fact that we're taking it off label, does that affect that question in terms of it being an orphan medicine?
No, I think that the good news, bad news is it going to be available at all? If it is going to be available in any way, then the community would have to decide: must a child with KCNQ2 mutation have a trial of ezogabine prior to enrollment in an investigational trial? Like, is that a hoop you must jump over? Same thing with carbamazepine. Is that a hoop you must jump over? Now, I'm not saying there's an answer to that, but that's the thing to think about with that. Ed.
Again, great to hear you. And following up on the last phrase, carbamazepine, I think, could you imagine that there could be a trial design where a family was, after informed consent, given a choice of carbamazepine, which in many centers, or an equivalent, IV phenytoin, oxycarb, one of those three is now often the first-line drug, and there's pretty good experience that for maybe not all, but for many patients, they get good seizure control as neonates. There's also a sense that that may not be the ultimate therapy for development. Could a family be consented to a new drug versus that drug, or would they have to, quote, fail the sodium channel blocker?
Well, that's the question that would have to be answered. Now, there's no controlled data on carbamazepine. So the argument to skip it would be a compelling case series, no controlled data, so no high quality data, therefore it's unproven, therefore there's no effective therapies, go right to the investigational therapy. So you'd have to decide, and every study site would have to decide that question. A different type of epilepsy that is struggling with this right now, infantile spasms, has 2, maybe 3, depending on how you want to look at one of the treatments, 2, maybe 3 treatments that are okay. They work 40, 50%. And they have control, at least 2 of them have controlled data that they work 40 to 50%. So, new drug. Right now, the thought is you have to try both of the therapies that have been proven to work. So you're putting in a 4-week and then a washout and then another 4-week delay. You can't try something new until you've tried both approved therapies. Okay? Yeah, but then it's hard. You can't study things at onset then. So effective treatments are always, again, it all depends on if it's working for you, it's good. If it's working for you or not working for you, then it delays the process. So, all right. Thank you very much.